The observed preference for protein-rich diet in Proceloleavis isopods. In this experiment, dairy cow isopods are used. They're called Proceloleavis, provided by the Bio108 lab. Before the experiment, they live in a moist soil container because naturally they prefer moist soil and a dark or dim environment. They are usually feed on leaf litter and dead organic materials. They are arthropods. They have segmented body with chitin exoskeleton. They also has a lot of legs, and they has dark dots on a white body, average around 1 centimeters long, but the size varies a lot. This experiment fed them apple, orange, liver, or potatoes to test their food preference. And our hypothesis is that isopods prefer liver because liver is rich in proteins. Isopods under humans are usually fed with plant debris, so we hypothesize that they may need more proteins provided by liver and over the sugar-based apple, orange, or potato.
So for our experiment, we wanted to see what dairy cow isopods, officially known as Proceloleavis, actually prefer to eat when given a choice. We set up a simple but effective experiment using a large petri dish as our arena. We tested 4 different food sources: apple, orange, potato, and raw liver. This gave us a good mix of sugars, carbs, and proteins to work with. Our experiment was split into 2 separate trials using 20 isopods for the first run and 15 for the second, with each trial lasting 15 minutes. For our data collection, we gathered both quantitative and qualitative info. Quantitatively, we recorded total visits to each food source by scan sampling. This method was used as it is nearly impossible to track 20 identical fast-moving isopods in a single petri dish. This gave us a better way to observe the attractions towards the different food sources. Qualitatively, we kept an eye on their actual behavior, like whether they were passing through or if they stayed to actively feed on a specific source. To keep the experiment fair, we kept the room dim, quiet, and constant in temperature to avoid stressing them out, and we reset the whole setup between trials to make sure the second group had a clean starting point. This is an annotated photo of our experimental setup.
The data shows the total number of visits to each food source over 2 15-minute observation intervals. As we can see in Table 1, in both trials, liver attracted the highest number of isopods with an average of 16.5, almost 4 times more than the amount potato attracted. The preference for liver remained dominant and even increased in the second trial, ranging from 15 to 18 isopods attracted. The potato showed a consistent level of interest with an average of 4.5 visits and a narrow range of 4 to 5 visits across the 2 trials. However, apples and oranges showed negligible attractions, with oranges recording 0 isopods in both trials, as we can see in Figure 1. The data indicates a strong positive correlation towards animal-based protein compared to plant-based starch and sugars.
Our results support our initial hypothesis that dairy cow isopods prefer protein-rich foods. As shown in our data, liver consistently attracted the highest number of visits, while carbohydrate-rich foods like apple and orange showed little to no attraction. This preference can be explained biologically. Protein plays a key role in arthropod growth and reproduction, and previous research done by Lardis and others in 2004 suggests that higher protein in isopods' diet can increase offspring production and growth rates. This likely drives isopods to prioritize protein-rich foods when given a choice. However, one limitation in the experiment is that we were unable to track individual isopods, so repetitions in counting likely happened. An improvement would be separating isopods or using marking or video tracking methods to produce more accurate data. Future research can investigate how environmental factors, such as moisture or light conditions, influence food preference in isopods. This is important because environmental conditions vary naturally, so understanding their effects would make findings more applicable to real ecosystems. In conclusion, our findings show that isopods strongly prefer protein-rich foods, which likely reflects their biological need for growth and reproduction. This helps us better understand their role in nutrient cycling and decomposition within soil ecosystems.